Hans, for the kind introduction and for the invitation to join this Monday webinar series of iNEXT-Discovery. And we are really happy to present today the work that we are doing in collaboration with the structural biology research infrastructures iNEXT and Discovery and INSTRUCT in the field of the fragment-based drug discovery. I would like to start just introducing EU-OpenScreen. So EU-OpenScreen is the European research infrastructure for chemical biology and early drug discovery. And our main goal is to develop chemical tool compounds, so-called probes, for basic and applied research. As infrastructure, we provide access to technologies, for example, compound screening platforms, but also access to resources like compound collection with different libraries. So our main library is a general purpose diversity library with 1,100 commercial compounds, but also we offer smaller non-bioactives, repurposing libraries, or in collaboration with the structural biology research infrastructure, we offer access now to a fragment library. EU-OpenScreen offers access to expertise in assay development as well, high throughput screening, and also chemistry for hit-to-lead optimization, but also access to fair data sets and training activities. We work together with users from both academia and industry. In our mission, we are supported by 8 countries, and we were established as a European Research Infrastructure Consortium in mid-2018. Our headquarters is based in Berlin, in Germany. Here an overview of our countries and our partner sites. EU-OpenScreen is a distributed infrastructure, and our partner countries are highlighted in this image in orange. And we have then a network of partners in these currently 8 member countries. We offer access to 3 different partner site categories. We have 17 screening platforms and also 6 chemistry groups that we help then the users to optimize the compounds that can be identified within the screening project and to develop them further, optimizing them in terms of potency selectivity. We have also one site that is hosting our database, which is the Institute for Molecular Genetics in Prague, as the screening data will all be made publicly available. Just a couple of words about how do we operate. So this is a scheme about our operational model. We usually work with external scientists, and Eric, the central office, manages the access to external scientists and bring them together onto our associated screening platforms and chemistry groups. As EU-OpenScreen, we operate a jointly used compound collection that I will describe later. This is in general our main users. So we support biologists, chemists, and data scientists, and from Europe and beyond. So if you want to screen a compound with us, if you have a biological question to answer, then the external scientist will ideally come with an assay which can be used for screening, perhaps in 96 well plate format. So this assay can then be transferred into one of our screening platforms. This will involve optimization and miniaturization of the assay and then the screening. We also can support users in assay development when it's needed, and because we have a distributed network of screening platform, we can offer a variety of different readout technologies. And after the screening, we have our chemistry groups that supports the user into the hit-to-lead optimization process. We work with chemists in the way that we are currently sourcing compounds from European and international chemists, which will then included into our European academic compound library. And this library will be then screened against a number of targets in screening campaigns, and also characterized by bioprofiling assays. So it's quite a benefit for the chemists because they can then identify novel bioactivities for their compounds. We also provide access to our data sets through the ECBD. So as I said, we make our primary screen data available in an open access database. We are committed to verification of data, but we offer also an optional embargo period up to 36 months. So users have the time to prepare a publication or to file a patent. I would like to introduce you our EU-OpenScreen libraries. because it will be closely related to our fragment library that we designed together with the structural biology community. So our main library is the diversity collection, which consists of commercially available compounds. It is sourced, maintained, and quality controlled in Berlin and shipped then to our partners for screening projects. It contains approximately 96,000 compounds with unbiased chemical diversity and have been designed by 5 academic computational chemistry groups from our network, which decided and discussed different design principles with the ultimate goal to build a highly diverse compound collection. Within the European Chemical Biology Library, we offer also a set of pilot library, which is composed of bioactives compounds. This is 2,464 compounds, which have been selected with a strong emphasis on target coverage and selectivity. But we do also offer a small set of representatives compound of the diversity library. So if a biologist comes to us and wants to prepare a proposal, we offer the pilot library in order to validate the assay and generate the first data that can be then used for project applications. With the idea always that at the end we would like to screen to propose to the user to screen the full set of the big library. As I just mentioned before, we are growing the European Academic Compound Library. So compounds are collected from chemists in Europe, but also from international chemists, and this number will be growing over the next year. But our target is to source around 40,000 compounds. The compounds will then be included in our screening campaigns at partner sites, and if a hit is found, then the IP stays with the user, with the chemist, and the IP rights can be discussed then with the assay provider. This will be the main topic of this presentation. So just recently we designed our new fragment library, which is composed of a set of low molecular weight and ultra low molecular weight fragments, and this work has been run in tight collaboration with the iNEXT and iNEXT-Discovery sites. I will describe this library in detail just in a second. Yeah, all the structures and all the QC data of the libraries are then available to the broad community through our European Chemical Biology database. Yeah, so this is an introduction about the fragment-based approach. So I would like to give you just a bit of background. We initiated this collaboration with the EU-OpenScreen and iNEXT-Discovery Instruct site, especially because we had the opportunity to get some funding through our European EU-OpenScreen Drive project, which foresees the extension of EU-OpenScreen capacity in fragment-based screening. The idea so is because the fragment-based methods are widely used to identify starting points in drug discovery and the generation of tool compounds for chemical biologies. So the basis of the fragment-based approach lies in fact in using smaller libraries, so only hundreds to thousands of compounds of low molecular weight, to identify then low potency, high quality leads. These fragment collections, which contain a lower number of compounds, that may achieve then similar coverage of the chemical space as the significantly bigger libraries required for traditional high throughput screening. Since the binding affinities of these libraries of the compounds are quite weak, we need more sensitive technologies to determine the binding. And usually, so to determine these ranges of micromolar to micromolar scales of activities, we need to use techniques like NMR and X-ray and X-ray crystallography. Following then the initial ligand identification, structure-informed design and fragment hit evolution can provide drug-like leads with high affinity and specificity. So it becomes apparent that the interplay between screening methods, structural methods, and medicinal chemistry require the collaboration of the two different communities, so the structural community, structural biology community, and the chemical biology community. So in such collaboration, iNEXT and the Instruct consortia that you know very well have automated pipelines for structural-based fragment screening in place, using a range of biophysical and structural methods, and they are currently offering them as a service to the structural biology community. In this joint collaboration, how we can complement this expertise, in fact, EU-OpenScreen joined the iNEXT-Discovery partners in the development of a new fragment library. We can then ensure the quality control, the maintenance, and the sustainability, but also support the structural biology community in follow-up studies of fragment hits, and also in fragment evolution. In fact, research groups usually performing the fragment library screening often lacks of this in-house expertise, and here is where we could support them with our medicinal chemistry support. So here I would like just to show you the EU-OpenScreen fragment library. So as I said before, it was designed by two of our chemoinformatics groups, more specific by Andrea Zagliani at the Fraunhofer Institute and Jordi Mestres. But it derives, so the main feature is that it derives from the fragment space of our ECBL. So it has been so designed that to facilitate, in fact, the follow-up with a rapid selection of parent molecules in the high throughput in our EU-OpenScreen high throughput diversity library, where, of course, the mini frags and the fragments are substructures. The library contains also a subset of fluorinated fragments, 131, which are then suitable also for fluorine NMR experiments, and it is consistent with the rule of 3, which is usually applied for the design of fragment libraries. This library has also been QC'd at the EU-OpenScreen Central Compound Management facilities, and then shipped to some of the iNEXT partner sites and the EU-OpenScreen partner sites. So yeah, within the EU-OpenScreen Drive projects, we set several goals to achieve that we would like to achieve during the project lifetime. And one was the design and the assembly of this fragment library, but also to monitor the performance of the library. And this will be possible if we maximize the usage of the fragments within this collaboration. Moreover, we established within this project a medicinal chemistry consultation service where user can then get advice on the hit follow-up studies. Just to give you an overview, so currently we ship the library to different facilities. So the library is available at several EU-OpenScreen Drive facilities, mainly for biophysical experiments, and at 5 iNEXT-Discovery Instructoric sites, which are mainly high throughput crystallization laboratories and NMR facilities. A bit of information of how you can assess our library. So if you are a researcher and you would like to assess the fragment library, then you will have, as we are two different networks and we are currently establishing the procedures on how to collaborate, you will have to follow dedicated access procedures. So in case that you apply for iNEXT, they have very well defined access procedures there, but still you will have to deliver some information to EU-OpenScreen. So we will ask users to provide key information about the projects like title, target, screening method that needs to be used. But more important is that we will need some information at the end of the project. So the feedback at the end of the project, we require some information about the hits, the hit rate, the number of hits, but also more information also about the structure, ligand efficiency, so on. We put in place We put in place forms to collect this information. So we are starting to work on the procedure for the users. We also ask the users, and in collaboration with their facilities, to make sure to upload the data in open access databases. And we also here offer an embargo period of up to 36 months. Here, just a representation. So how this joint workflow with the iNEXT site looks like. So practically, you can screen our fragment library at iNEXT and EU-OpenScreen partners using different technologies. And then once you will get you will identify your hits and get some structural information, you can follow up with our chemistry groups. So the main idea here is that our chemistry consultant, let's call them consultants, will follow up, will give you a proposal on the follow-ups from the European Chemical Biology Library, so our big commercial set. They will also propose commercially available fragment hit analogs, also synthetic fragment follow-ups based on the synthetic feasibility of the fragment, strategy for fragment merging, and the fragment grow analogs if the structural information are available, and also estimate some workload for the synthesis, and then try to match with the chemistry side that is more suitable for the project. I will start to conclude. I wanted just to show you that for the hit to lead optimization part of the project, you can always contact one of our 6 chemistry partner sites. So all our laboratories are multidisciplinary research groups. They have an extensive experience in medicinal chemistry field, including structure-based drug design, the hit to tool and lead optimization, and they work in a vast number of research areas, diseases, and targets. I hope I gave you an overview about EU-OpenScreen. If you have any questions, you can always contact us. I'm Tanja Miltic, project manager at EU-OpenScreen, but also you can contact Aigars Jürgensons, who is the head of the Organic Synthesis Methodology Group at the Latvian Institute of Organic Synthesis, and he's in drive our work package leader for the extension of our service portfolio in a fragment-based drug design. I will let now the word to Kristaps, and he will present you a practical work where we have used our fragments library designed together with iNEXT-Discovery sites into a screening for SARS-CoV-2 targets. Thank you so much. I will stop to share.
Okay, hello everyone. I'll put my screen on. Okay, I hope you can see it now. So I'll start with a quick introduction to the Latvian Institute of Organic Synthesis. So this is a public research institute. So this is a public research institute located in Latvia, in the capital of Latvia, in Riga. And we are the EU-OpenScreen medicinal chemistry partner site. And this, as Tanja already mentioned, means that we offer medicinal chemistry consultations for the projects that have been approved by EU-OpenScreen. And we are also the leader of the EU-OpenScreen Drive project, work package 4, on extension of capacities into fragment-based screening. Here, this is an overview of the EU-OpenScreen fragment library characteristics. I think I will not go into details here. It is, of course, rule of 3 compliant library enriched with low and ultra low molecular weight fragments, as you can see here. But the very nice feature of this library is that it is derived from the fragment space of the EU-OpenScreen small molecule diversity library. And this means that for every fragment, we should be able to find follow-up compounds in the 100,000 compound drug-like library. This is how it looks. Basically, it's formatted in these 96 well plates at 25 microliters, and the compounds are dissolved in DMSO D6 at 100 millimolar concentration. And now I will present this case study where we used this library to screen it against the NSP14 protein of SARS-CoV-2. So in July last year, we decided to join the global efforts in the fight against COVID-19, and particularly the efforts of the NMR community, and to take part in the Latvian state research, Latvian state COVID-19 research program. So we chose this SARS-CoV-2 mRNA cap methyltransferases, NSP14 and NSP16 as targets because previous studies on SARS-CoV-1 had shown that NSP14 is an exonuclease and guanine N7 methyltransferase, which is required for viral mRNA translation, while NSP16 is a 2'O methyltransferase, which is important for evasion of host immune response. So these are both validated drug targets against coronaviruses. And both of these enzymes use S-adenosylmethionine, or SAM, as a methyl group donor for the methyl transfer reaction to the mRNA. And today I will talk about fragment screening against NSP14 because with NSP16 it has been difficult for us to use the fragment-based approach due to poor protein stability. Here on the right, you can see the structure of NSP14 from SARS-CoV-1. It comprises two domains. The exonuclease domain is shown here in green, and the N7 methyltransferase domain is shown in blue. The protein has several binding sites. The exonuclease domain forms a complex with NSP10, shown here in red, but NSP10 is not required for the methyltransferase activity. While this methyltransferase or MPase domain binds SAM, so S-adenosylmethionine, and the mRNA. Additionally, this exonuclease domain can also interact with the RNA-dependent RNA polymerase, NSP12. But we chose to target the SAM binding pocket with fragments because development of selective viral methyltransferase inhibitors has been a challenge using a more traditional substrate-based design approach. And actually, we have also previously used this SAM co-substrate-based bioisosteric design approach and have discovered new nanomolar inhibitors, but they all were not specific for the viral over the human methyltransferase. So the fragment screening was done using nuclear magnetic resonance spectroscopy, or NMR, and we screened the library in cocktails with 8 fragments in one cocktail at 250 micromolar concentration in phosphate buffer. The protein concentration was 12.5 micromolar, and to identify binding to the S-adenosyl methionine binding site, we used a competitive inhibitor, cinefungin, also at 250 micromolar concentration. And the samples were prepared in 3 millimeter NMR tubes. The equipment that we used was a Tecan Evo 100 pipetting robot, which was equipped with an NMR tube rack to pipette into NMR tubes. And we used a 600 megahertz Bruker Avance Neo spectrometer equipped with a QCI-F cryoprobe. And this was also equipped with a sample case sample changer. And then the experiments that we ran was were a water loxy and a T1 row or CPMG experiment. And then the whole screening was done in a way that we first recorded spectra of the reference samples or cocktails in the absence of protein. Then we added protein, recorded the experiments again, and if there were binders among the 8 compounds, then we added this competitive inhibitor to identify compounds that bind to the SAM binding site. And the whole screening required an effort of about 1 person month. And here on the right, you can see some examples of the spectra. So this is just a validation of the method with S-adenosyl methionine, and we can see the change of signal sign in the water loxy spectrum and the decrease in signal intensity in the T1 row experiment, which confirms the binding of SAM to the protein. And here, this slide now summarizes the results. So in the end, we had a pretty high hit rate of 13%. And the criteria that we used to identify hits were that we observed change in the sign in the water loxy spectrum and signal intensity reduction of about 20% in the T1 row experiment. And the relatively high hit rate is probably related to the large number of binding sites on the protein surface. But however, only 3 fragments were identified as specific SAM pocket binders. And now we are looking into the follow-up studies with these 3 fragments. So we are in the process of testing inhibition, and we already have some preliminary data showing that there is measurable inhibition for these fragments at 100 micromolar concentration. And then the next step will be, of course, cherry picking and testing of the follow-ups from the EU-OpenScreen small molecule diversity library. And we have also performed the screening against this NSP14 with another fragment library, the iNEXT library. So there will be also the possibility to compare the results. Actually, I can tell you already now that the hit rate is similar. It's a bit lower, about 10% for the iNEXT library, but the number of specific binders to the SAM binding pocket is very similar, also around 2 or 3. And with this, I would like to conclude and acknowledge the people involved in this study. So Anna Bulla was the one who performed the fragment screening. Gins Kalniņš prepared the proteins that we use. And Kaspar Stars was our project leader. And I would also like to acknowledge Aigar Sirgensons, who coordinated the obtaining of the fragment library from EU-OpenScreen drive. And with this, I would like to thank you for your attention. And I look forward to your questions.